Today I want to talk about Pacific lamprey and the White Salmon River Basin after the breach of Condit Dam a number of years ago. Folks in our office have been working in this basin for a number of years, some of them before my time, so there's a lot of folks who've thought a lot about this. If you see this picture here, this is an extreme close-up of a larval Pacific lamprey. These are really cool fish. See, this is the oral hood here in the larvae. It looks much different than what these things eventually turn into as adults. So a quick roadmap here, overview, give you a little bit of background on the problem. The applied science bullet here could also be termed as the methods. Sort of with the theme of the meeting, I wanted to highlight a couple of the different science tools and products we've brought together to make this work as strong as we can. I'll get into the study details, the nuts and bolts of what we did and what we found, and then finally a little bit of a look towards the future. So just to orient us, the White Salmon River Basin, a lot of you folks know where it is. It's in south central Washington, and the headwaters here is up at Mount Adams. That's a source, and then the mouth is down here with the Columbia River at Bonneville Reservoir. Essentially dumps into Bonneville Pool across the river from Hood River. And Condit Dam was not very far upriver here, 3 kilometers or so. So it really had an effect on a large portion of the basin. And so you can see an aerial photo here of here's the dam and the small reservoir that it impounded as well. And so, I mean, the background, Condit Dam was there for a long time. It was completed somewhere around 1913. It had fish ladders. They didn't last very long. They got destroyed in a number of years. And the dam essentially blocked fish passage for close to 100 years to anadromous fish. The dam came out in 2011. And essentially, this talk is really about before and after for lamprey in the basin. It's that simple. So some of our overarching questions had to do with what's the distribution of lamprey prior to and after the removal of the dam? Can the presence of western brook lamprey help guide us to places where Pacific lamprey might recolonize? So western brook lamprey are the native non-migratory lamprey species that we have out here that co-occur with Pacific lamprey, and we already had prior knowledge that there were western brook lamprey up in the basin. So having a better idea of what parts they occupied might key us in on habitats which would be suitable for Pacific lamprey as well. How much of the basin is actually accessible to recolonizing fish? It's a steep, canyonized system in a lot of places, a lot of waterfalls, so there's probably a lot of areas that actually aren't accessible to anadromous fish. And some questions about habitat. I'm not really going to talk about that today. So more specifically, objectives were to evaluate and compare occupancy by larval Pacific lamprey and western brook lamprey before the removal of the dam and after the removal of the dam in a number of different habitats. In wadable areas upstream of the dam, in wadable areas downstream of the dam, and also in so-called river mouth or delta, that place where the white salmon meets Bonneville Pool. So a couple different sorts of habitats we're interested in. And some of the science tools we brought together here, and I'll explain a lot of these in more detail as I progress through the slides, but we have specialized sampling technology, both in wadable streams and in larger boatable areas. Techniques, tools that have been developed for sampling larvae. We employed an occupancy-based sampling approach. This is sort of new to some of us. It's been around for a while. I think it's new, even newer to fisheries folks. It's a different way of thinking about sampling, and it's a presence-absence-based sort of approach, and it has a lot of promise for certain situations. It can be very useful. And couple that with a probabilistic design that we're using probabilities, and at the end of the day, we can say something about presence and absence with some sort of level of confidence about that. And finally, the genetic tools that we have over the years, collaborating with some of our partners to understand the genetics of these species, that's a big part of this story. So brought in a lot of different tools. So let's talk about the where. Where are we going to sample? So one of the starting points we've used in the past for lack of really good information about to be able to pinpoint the habitats that Pacific lamprey might use, we've used steelhead intrinsic potential because we feel like more or less steelhead and adult Pacific lamprey might utilize similar looking habitats. And so and this is an approach that was sort of used in the early phases of the Pacific lamprey conservation initiative as well to start guiding us to where we think that they could be. So in this figure, you see there's not a lot of the basin that really gets much of a score. A little bit of the main stem of the white salmon, and this basin is Rattlesnake Creek. It's a kind of a high score for steelhead intrinsic potential. So it starts guiding us into basins that we might want to look for lamprey in. You know, up here, there are barriers, and the thing with Pacific lamprey is they can climb. We don't exactly know what is a barrier and what isn't a barrier. So there are questions about could there be areas up here that they might be able to get to. And so once we sort of have a set of sub-basins that we were interested in, we next wanted to figure out how do we select our sites. In the wadable areas, a sampling reach is a 50-meter reach. So in this example, we're looking at Buck Creek watershed. We use a GRITS approach, and that stands for Generalized Random Tessellated Stratified. It's sort of a funny acronym to just describe it's random and spatially balanced. I mean, that's the utility of it. So you divide up all your possible stream reaches, and the GRITS procedure randomly orders those and assigns them numbers. And so in this instance, I've shown about the top 40 reaches. And say we decided we need to sample 5 reaches. So we'd go through and find, you know, reach number 1, reach number 2, et cetera, until we hit our site. In the river mouth areas, it's more of a two-dimensional problem because we're sampling from a boat, and our sample site is a 30 by 30 meter quadrat. So in this generic example, we divide all that into all the possible quadrats. That's our universe of potential sampling sites. Again, we apply the GRITS procedure. It orders all those in a random way, and we sort of have a starting point of where we want to look. And then finally, is how hard do we look? The key question here is, what do we do when we go out and look for the animal and we don't find it? What do we say about that? Is it really not there? Is it so rare that we didn't find it? And so we use models of detection probability to help guide us in that sampling effort to give us a desired level of confidence. So in this situation, in the wadable streams, we used a conservative detection probability, very low, of 0.07. And essentially what that gives you on this curve is a predicted number of sites that you need to sample. So in this case, 21 sites. And basically what that means is if we go out and sample 21 sites and we don't find any lamprey, we're 80% certain that they're not there. So we're able to say something with some level of confidence about it. That's really the only point in all this. And the same in the situation in the river mouths, we had some previously determined detection probabilities from some work in the lower Willamette. And essentially to get that desired level of confidence, we needed to do 17 quadrats. We doubled that to 34 to be conservative, to take into account that these are different habitats and the detection probability might be different, and so we wanted to maybe capture that. So how do we get them? In the wadable streams, we use backpack electrofishing. It's got specialized settings for larvae. I call it tickle and stun. It's the tickle charge. It's not just a wrestling move. It's good for lamprey as well. But the tickle charge gets them agitated up out of the sediments. That's where they are. They're burrowed in the sediments, filter feeding. And then so you agitate them up out of the sediment, and you see fish, then you hit the stun charge, and it immobilizes them, and you can net them out. We identify the fish using external characteristics. We take a fin tissue clip for genetic confirmation. At a certain size, below 50 millimeters or so, you can't tell the species apart. The external characteristics don't work, so you really need genetic confirmation. The deep water electrofisher is its own sort of crazy contraption put together by folks in the Great Lakes who work on sea lamprey. But essentially, it's an electrified suction dredge. So here's a schematic. Here's the business end of it right here. It's kind of a cone, and it gets lowered to the bottom of the river. So here it is sitting on the bottom of the river. It's electrified, so it has that tickle charge, gets the gets the fish up out of the sediment, and then they're brought up through the tube and filtered through a basket. So here's what it looks like hanging over the edge of the boat. Here it is in action. Here's Christina Wu running it, filtering, and you can see it's not a really deep spot here, but it's sitting on the bottom. And then here you can see we captured a larvae in the basket there. And so let's look at some of the results. What you see on this, every dot indicates a sampling location. A red dot indicates a place where we found no lamprey. The purple dots represent dewatered areas. The white dots represent places where we found western brook lamprey only. And the diamond is a spot where we found both Pacific lamprey and western brook lamprey larvae. So we learned a lot here in this pre-sampling. First of all, we learned a lot of these places were inaccessible. Either they dewatered and they were ephemeral. We ruled those out as possible places. Some of these basins I don't think are even able for fish to get in because of waterfalls and everything. And so basically we're ruling out a lot of places, so when we return we're going to be much more efficient in targeting places where we think they can be. So what we found here, we had western brook lamprey up in Trout Lake Creek and western brook lamprey in northwestern reservoir, the impoundment, and then both western brook and Pacific lamprey below the dam, and that's further corroborating some anecdotal information that we had that adult Pacifics were trying to head up the white salmon and maybe spawning up there in that limited reach that they had before they hit the dam. There's some of the waterfalls. Like I said, we don't exactly know what a barrier is, but I think sometimes we can say with some level of confidence what is. And so post-dam removal, we sampled a lot less places because we had that information. We still went to Trout Lake Creek. We went to Rattlesnake Creek, and that was important to us because that got such a high score of that from that steelhead intrinsic potential. So we were curious if Pacifics would head up that way. And then Buck Creek and Mill Creek. And so again, in Trout Lake Creek, we found only western brook lamprey. In Buck Creek, we did not detect any fish. In Mill Creek, we found only western brook lamprey larvae. Rattlesnake Creek, we detected one larvae in one reach, and we had never detected any fish there before, so that was, again, showing us there's at least habitat for burrowing to happen. And then here's the exciting slide. We had 4 reaches where we detected Pacific lamprey larvae above the dam location. This is last year. 3 of those reaches are actually where Northwestern Reservoir was, so they're in the old footprint of the reservoir. This reach is actually above where the reservoir was. This was pretty exciting to us, and here's what they look like. They're really small. We couldn't identify this, and we sort of, because we hadn't identified any Pacific lamprey, we didn't think there were any there. We sent in the genetics and sort of forgot about it, and I got the results back, and I started looking at this, and we had genetic confirmation of 13 specimens in 4 different reaches came out as Pacific lamprey. So sure looks like we had some sort of recolonization, or at least a spawning event or something, and some young ones were produced. They were all between 25 and about 50 millimeters, so also very small, and what we might expect if they're sort of the progeny of a spawning event. And then quickly in the river mouth area, so in 2011, before the dam went out, we sampled a whole bunch of places. We didn't find any larvae. And there was really no habitat there. It was deep, it was swift, it was bedrock, boulders, and it was sediment starved. And that's because that dam had held back that sediment for 100 years. So after they blew the dam out, all that sediment got flushed down the river and out into the pool. And in the very next year, we detected a western brook lamprey in that. And there's a sandbar and everything there, and it's a new windsurfing place. And the next year, we got Pacific lamprey and western brook lamprey. So there's new habitat that didn't even exist, and now you have larvae that are rearing in that. So that was kind of cool to see that dynamic. Kind of to wrap it up here, I mean, it's a really neat opportunity to sort of examine a natural recolonization. One of the things that really makes me happy about this occupancy-based method works. We're real efficient. It's not hard for us to detect these fish. We don't have to go to a lot of sites, and we feel pretty solid about this presence-absence-based technique. The techniques are flexible. I showed you a couple different ways that we applied it, and it's even more flexible than I described, depending on what your question is. And again, the genetic tools were a huge part of the story. So into the future, we have other questions about range expansion. I mentioned Rattlesnake Creek, Beezy Falls. I wonder if that's actually a barrier. We've thought about doing some surveys above that. But so this is sort of brand new stuff. We haven't really even talked exactly through what the next steps might be. So that's the end of the trail, and I'll take any questions. Except the genetic tools were eDNA. No, we haven't used that. It's just that we developed the markers to identify and distinguish the two genera, Entosphonus versus Lampetra. That was something. That was a tool we didn't necessarily have until 6, 7 years ago. He asked if the sediments that got flushed down the river and into the pool, if I think those should persist. I don't really know. It's very dynamic. It looks different every year. You know, other similar rivers like the Hood River and the Klickitat have sort of these extensive delta areas. The ship channel kind of winds up that way. And I wonder, at one point, could that be something that the Corps might want to address? But it doesn't look like it's out there yet. And so, more specifically, I don't know if it will persist, but I think it will.
So when you say address, you mean?
Yep. Which they do in other places where they have sedimentation projects.
In your samples, can you assess from that if there might have been different age classes of fish?
In the Pacific lamprey larvae that we detected,
Pacific, not necessarily the West.
yeah, they were about 25 to 50 millimeters. It's just assigning age from size is a dicey endeavor with these. It's the 50 millimeter thing makes me wonder that we may have maybe two different. It's a pretty broad range, I would say, that we may have a couple different years of progeny out there, but it's speculative at best.
Jeff, to what degree now that we have more dam removal proposals out there, to what degree are some of the lessons you've all learned in terms of when and how and why to be looking for lamprey recolonization being folded into some of those next phases of pre and post dam removal monitoring?
I'm not sure I quite get the question.
So basically just wondering, for either your office or partners you're working with, are there lessons learned from your work that are being incorporated into pre and post dam removal monitoring for lamprey in other areas? I was not part of the years of sort of discussions and things. I mean, 15 years or more of talking about Condit Dam, and I know that we always had a voice in those discussions. That wasn't me, so I don't really have that perspective. It's kind of a new, I came in at the fun part right before it got blown out and got to do work. So I think there are probably others in this room that might have a little more expertise there.